I'm Fred Gerard. I'm the CEO of Graybug Vision. Thank you very much for OIS to invite Graybug today to present our progress in drug delivery. You will find here our forward-looking statements, and in a nutshell, Graybug works on long-acting treatments for vision-threatening diseases. We have a number of candidates, and as you can see here, one is clinical stage, GB102, our lead asset, a pan-VEGF inhibitor for the treatment of wet AMD, which we intend also to develop in DME. We have the same molecule in a once-a-year formulation that we intend to develop for diabetic retinopathy called GB103, as well as a proprietary prodrug beta blocker called GB401, which we intend to develop for every 6-month injection in primary open angle glaucoma. Our technologies are well protected to 2039 for retina and 2041 for glaucoma. And we have a number of milestones. The next one is the readout of an ongoing phase 2b study, which will read out in the first half of 2021. And we plan to initiate 4 new clinical trials in the second half of next year. On slide number 4, this is how our proprietary technologies present. We inject in the eyes a suspension of microparticles with a small gauge needle, and in contact with the vitreous, these microparticles come together, aggregate, and form a depot from which a drug will be released over a period of time. These depot itself, these polymers, hydrolyze over time and disappear from the eye without leaving any presence of byproducts. We work on differentiating mechanism of action. On one hand, we have a pan-VEGF inhibitor in retina. We have a proprietary prodrug in glaucoma. We can tune the durability of these polymers from a few weeks to a year, as well as control the drug elution from these polymers in a very good way. On the hand, now have some photographic evidence of what we can do to present to you. Our technologies are versatile. We can actually use them for a number of molecules. And of course, we design everything we do with safety in mind. And here you can see our pipeline on slide number 5. So I'll start with the retinal program. So first, a quick introduction of sunitinib. Sunitinib is a good candidate for a pan-VEGF inhibition. As you know, traditionally existing products block circulating VEGF-A from binding to receptors 1 and 2. We decided to go with a different approach, with a small molecule approach, blocking all VEGF receptors. And when these receptors are blocked, basically it will antagonize the effect of VEGF A, B, C, and D, which, as other companies have demonstrated, actually might provide some additional benefit to the blockade of VEGF A only. Incidentally, sunitinib is a potent neuroprotectant. It's a dual leucine zipper kinase inhibitor that has demonstrated preclinically some interesting efficacy in neuroprotection. We conducted a first-in-human study called Adagio. It was a safety study looking at 4 different doses of GB102 administered to patients that had received previously at least 3 injections of anti-VEGF, short-acting anti-VEGF, and these patients were given either a dose of 0.25, 0.5, 1 or 2 milligram of GB102, only 1 dose, and were monitored for 8 months. Basically, these patients did well. We achieved the primary endpoint of safety or tolerability. As you can see here, we had no SAE, no ocular SAE or dose-limiting toxicity, and most patients did well in that study. The most common adverse event was actually presence of microparticles in the anterior chamber. As you can see here, this one single injection of GB102 was able to control retinal thickness and visual acuity over in these patients. And actually 7 out of 8 patients did not require any other supportive therapy in the first 6 months of the study. Actually 4 out of 8 of them going to 8 months or beyond without any additional anti-VEGF. Because of this presence of particle in the anterior chamber, we decided to further optimize the manufacturing process of these microparticles. And we conducted a small open label safety study in macular edema patients with the same type of patient population, different disease, but these patients were also pre-treated with at least 3 injections of short-acting anti-VEGF, got one injection of GB102 and were monitored monthly for 6 months. And we tried two different doses, 1 and 2 milligram of GB102. This is how this enhanced formulation presents, and it's injected after a reconstitution in the eye over 5 seconds at a 6 o'clock position, as you can see here in a glass eye model. And as you can see, these microparticles get out of the needle and start aggregating immediately when they get in contact to the vitreous. And this is how these particles look like in the eye. At baseline, you can see for the first time, we have these fantastic pictures in human eyes. You can see at baseline, the depot forms immediately, and then the depot stays in the eye, collapses on itself all the way to 6 months, which is the bottom right picture, where you can see a little bit of polymer left, bright orange, still at 6 months, demonstrated that some sunitinib is still left in the polymer at that time. The GB102 1 milligram was well tolerated. We still observed some presence of medication in the anterior chamber with the 2 milligram, which was not as optimized as the 1 milligram, and we decided to stop that dose. But overall, the 1 milligram was well tolerated and controlled the vision and retinal anatomy of these patients. We are conducting currently the larger phase 2B in wet AMD called Altissimo. This is a double-masked randomized study comparing GB102 to Eylea, given bi-monthly. And you can see here, we had originally two arms of GB102, a 1 milligram and 2 milligram arm. The patients that were in the 2 milligram arm were re-injected at 6 months with GB102 1 milligram, and we expect the results of that study in the first half of 2021. I'll just say a few words on our glaucoma program. Our glaucoma program, as I said before, is an inactive prodrug embedded in the same type of polymer as GB102. This prodrug gets activated when released, and you can see on the right-hand side the quality of the pharmacokinetics we get from that formulation. A proof of concept has been done in ocular hypertensive rats, demonstrated that the approach of injecting GB401 IVT could result in an IOP reduction similar to timolol eye drops. And you can see on the right-hand side, we looked at target tissue concentration over 6 months, which regardless of the dose was quite stable over 6 months. At no time point was the active beta blocker detected in the plasma. And we expect to start a first-in-human study in the second half of next year for this formulation. Thank you very much.